Today we will focus on the single omics or to the multi-omics. So I think among these proteomics technology, there will be the one that fits your research needs. Okay, the first, let's take a look at the overall background of the proteomics technology. So going from the genotype to the phenotype involves many biological processes. So at the DNA level, many epigenetic modifications occur, which affect the phenotype. Then the DNA is transcribed into the RNA, and the RNA is translated into the protein, and the protein perform their function. So during the life function process, the metabolites are produced. So all these processes will shape the final phenotype. So at a different molecular process, at the different molecular levels of the life, there are different research detections. So in the genome levels, we can maybe, for example, we can access the DNA levels, DNA copy number and screen the mutations. And for the RNA levels, we can focus on the mRNA expression and the interaction on the non-coding RNAs. So for the proteomics, we can explore the protein conformation and the protein expression level and the various modifications that happens to protein. So there also will have the metabolomics, and which can be further divided into the glycomics and the lipidomics, and the other areas focus on the different metabolites. So each omics fields will help us understand how the information in genes is transformed into the traits we see. So what is meaning the how we get from the genotype to the phenotype. And in the genetic central dogma, the DNA is transferred to RNA, and the RNA is then translated into the proteins. So among all the omics fields, you can see that the proteomics is very close to the final phenotype. So some studies have looked at the distribution of the mRNA and the protein quantities, so as well as the distribution of their correlation. So they comes to these conclusions. So in a single cell at the specific time, there is no reclusion between the mRNA and the protein levels. So this means that the mRNA and the proteins do not have a simple corresponding relationship at a given moment. So it also shows the complexity of the life process. So we can't just infer the protein conditions from the mRNA alone. We need to do more comprehensive, maybe the multi-omics research to deeply understand the biological process. So therefore, combining different omics fields can better help us understand the mechanism behind the biological process. So about the proteome, so the term proteome comes from combining the protein and the genome. It refers to the complete set of the protein expressed by a genome. So the unique biological information carried by the protein cannot be replaced by the genome or the transcriptome. So for example, the proteins have a spatial and have a temporal dependence, and the mRNA level do not cross bound to the protein expression level. And then the proteins also can be modifiable. And the post-translational modification and the isoform cannot be seen at the gene level. So in the next part, the protein-to-protein interactions are hard to understand at the gene level. So however, even though the proteins have these all replaceable features and are extremely important for analyzing the phenotypes, but the proteins are much more complex than the genes, than the transcripts. So it makes it's very important to choose a suitable technology for your research, as well as ensuring the maturity and accuracy of the detection technology. Okay, so the next, let's take a look at what proteomics solutions that BGI can provide. And in this slide shows all the omics services BGI can offer. You can see at every level of life activity, from the DNA to the RNA, from the protein to the metabolites, and even the multi-omics, we have corresponding services to meet the needs of various research detections. So besides offering a wide range of the omics services, we can also have many options for the instrument platform. So these include the high-throughput sequencer with the long and short read, and the mass spectrometers from different platforms. So all these support us in providing the fast, the accurate, the reliable omics data to our customer. So in our high solution MS platform, we have the Orbitrap series instruments such as the Astra and as well as the Time12 and the QEHF series platform. The high resolution MS platform is mainly used for the untargeted MS technology, like the proteomics identification and quantification, and also will use it in the post-translational proteomics identification and the quantification. So for our triple quant MS platform, mainly use the Q-Trap series instrument to meet the needs of the targeted MS product. Okay, so for over the past decade also, with our rich experience in the mass spectrometry technology, we has already published over 350 papers independently or with our customer together. So most of these papers were published in the major global journals. So with a total IF of over 2,000. And we have provided services for more than 7,000 projects. So in addition, we have participated in the human protein project and have already published 18 related articles. So the total number of the published paper has been gradually increasing. So based on our rich project experience, we have established a strict quality control standard, which is divided into these 4 main stages. So for the sample stage, we will check the sample information and evaluate the sample quality. And next, in the sample preparation stage, we will prepare the QC samples, the black samples, and the QA samples. And then in the QC quality control stage, we will use the black samples to check for the no observed impurity peaks, and we will use the QA samples to check the quality deviations. And we also use the QC samples to check the response value and the retention time deviations. So in the finally part for the data analysis stage, we will create the base peak chromatogram over the plots, overlap plots for the QC samples, and also we will have the PSA plots including QC samples, and also we'll have the CV distribution of all peaks in the QC samples. So there also will have the there also has the real-time monitoring process to detect and solve problems in time. So this whole all complete a strict quality control process will ensure the reliability of the experimental data. So what the specific protein services can BGI provide? So we know there are many protein regulatory events in the cell, and the protein refers to the complete set of the protein in the research subject. So at BGI, we can start the protein of the biological individual from different dimensions, such as the protein identification and the quantification research will help us to understand the composition and expression levels of the proteins. And for the modifications research, we can deeply reveals how the proteins are active or inactive through the modification. So first, let's talk about the protein identification services in BGI. So in this services, we have 3 kind of types of the samples. The first one is the protein in the gel spot and the gel band after you doing the purification and enrichment. And the second one is the protein obtained from the protein interaction experiment, such as the IP, the co-IP, or the pull-down, or the cross-linking experiment. And the third one is the protein from the tissue, like the cells, like the water and the feces. So if your goal is only to identification, not to do the quantification, so maybe you can choose our regular protein identification product. For the first 2 kind of sample type, you just need to send us the protein solution. But for the third sample type, in these samples, there will have more types of the proteins with the higher abundance. So we recommend our proteome profiling services for the identification. So, and you can send the cells, the tissue, and the other sample type directly to the BGI. And if your research requires more than just knowing what proteins are in the sample, you also need to quantify the protein expression, and you need to compare the difference between the different groups. Then I think you will need to the protein quantification products. So here we have 3 types of the technology for the protein quantification, which fall into the untargeted and the targeted. So for the untargeted technology will include the label-dependent technology, which we use the TMT. And for the label-free technology, we will include the DDA and the DIA. So for the targeted technology, the O-link is the main option, and the customized PRM targeted services will be a supplement. So the first one for the label-dependent protein quantification technology, we mainly use the TMT tags. So the tags is made up for a reporter ion and a mass normalization group and a reactive group. So different labeling reagents have the same molecular weighting, but their reporter ions will have the different molecular weights. So after the labeling different sample with the different reagents, we mix the sample, and then the labeled group are released in the collection cell, and the signals of different labeled group will appear. So based on the signal of different labeled group, we can get the quantitative information of the same peptide segment between the samples. And then this can be further converted into the quantification, quantitative information of the proteins. So the TMT allows us to label and to analyze the multi-samples in one experiment. For example, the TMT Pro 18-plex can handle the 18 samples at the same time, and this greatly improves the experimental efficiency and reduce the impact of batch effects. However, it also limits the maximum number of the samples we can test in one experiment. So if you have more than 18 samples for protein quantification, we need to mention the label-free based the MS detection technology. So in this technology, there has no limited on the number of samples, and it's currently a mainstream protein quantification technology. It has 2 detection models, the DDA and the DIA. So the DDA is based on the peak intensity information for the parent ions obtained from the first MZ scan. It only selects a few parent ions with the highest signal intensity for the fragmentation. So this selective collection leads to the randomness and the incompleteness. So for the DIA, after the peptide enter the mass spectrometer for the first MZ scan, all the parent ions within the select MZ range will be a fragment. And then a second MZ scan for the doctor ions is performed in the tandem mass spectrometers. So the DDA is more suitable for the less complex samples, and the DIA is suitable for almost any condition, which means the small to large cohort, the complex proteins, and the low abundance proteins, and so on. So because of these advantages of DIA, it's more widely used than the DIA model. And regarding the DI technology, there have some leading experts in the field of mass spectrometry and proteomics once it started. They joined handles with the 11 MS lab from different parts of the world using the same experimental and analyze the procedures to conduct a comprehensive evaluation of DIA technology. So the result shows that the different labs could obtain the high reproducible protein quantitative data using the DIA technology. And its reproducibility and sensitivity are very close to the SRM, which is the gold standard for the protein quantification. And this laid the foundation of the application of the DI technology in the large-scale protein quantification. And BGI was participant for the site 10 in this study, and the research was published in the Nature Communications in the 2017. So later, the BGI collaborated again with the 11 lab for 9 countries around the world to establish a set of standard procedures for the large cohort DIA protein quantification. So this ensures stable, the sensitive, the reproducible data across the multiple centers lab supporting the accurate research. Okay, besides the high quality methodology, the BGI is also at the forefront of using the instrument platform. We have introduced the thermal Orbitrap, the Ultra MS platform, which further enhances the performance of our DIA services. The Orbitrap Ultra instrument fully inherits the ultra high resolution, also ultra high sensitivity, and ultra high dynamic range of the Orbitrap instrument. So at the same time, it has an active new generation mass analyzer, which achieved the ultra high sensitivity without losing screening speed and high resolution. So on the right side, you can see BGI's actual test data for different sample types on the different platforms. You can see that the number of the identified proteins is higher on the Ultra platform. And when we're dealing with the meta samples, we also have the corresponding meta proteomics solutions. You can send the sample like the faces, the oils, the soils, and the waters to us, and we will detect the composition and expression of the meta proteins in the meta samples. So besides the special case of the meta samples, we also face the difficulties in some sample collection, and we also meet the limited sample volume during the actual sample collection. So if you have the macro samples, don't worry. We have also developed a special detection protocol for the macro protein samples. It can support the detection of as few as 1 to the 100 cells. Okay, all above are our untargeted proteomics detection service. So if your research goal is not to observe the overall proteomics situation, but you already have target proteins and need to accurately understand the expression of these proteins in a targeted way, then you can choose our targeted protein detection service. You can select a suitable panel from the Olink panels. So if there is no panel that meets your needs, we can also evaluate and provide a customized PRM detection to target your specific proteins. So about the Olink technology, it's a protein detection technology developed by the Olink. Its main principle is to link a target protein with a specific nucleotide sequence through the specific antibody. So then we will detect the nucleotide sequence to achieve the protein quantification. So this technology is widely recognized and used because its significant advantages like the high sensitivity and the high specificity. And now, sorry. And currently, the BGI can provide almost all Olink panels, such as the target series based on the qPCR quantification. Also, we can provide the Explore series panels based on the NGS quantification. And the application of the Olink technology mainly focus on the human or the mouse blood samples. It's quite common in the disease research and the large sample cohort study. And for the next part, it's for the post-translational modification refers to the process here. And If your purpose is to know the modification in the proteins, you can choose all these product. So after protein is produced on a ribosome, the chemical group will added into his amino acid residues. So its structure will change, all will be fragments. So this modification directly affect the key property of the proteins, such as the activity, the localization, and the interactions. So studies have shown that more than over maybe 50% of the proteins in the human body will undergo the post-translational modification. And currently, BGI can provide the detection for several major types of the protein modification. And since the expression level of the modified proteins is very low, we will enrich the extracted protein based on the cross-body modification. So at the same time, we also will add the evolute analysis of the modified proteins during the analysis stage. So besides the regular analysis, such as the modification size, the modified protein and the modification types, if you want to conduct in-depth research on the modification, we have also a product which is the nitrogen link intact glycopeptide proteins research protocol. So it can obtain the glycan structure and the glycosylated peptide segments and the cross-bonding relationship between the cross-guide and the classical state. So allowing we can do the comprehensive, we can do the in-depth and the detailed research on the glycoproteins. So in the terms of the data analysis, to make it easier for our customer to use the data, the BGI also developed a wide-based solution, the Dr. Tom system. So on this system, you can perform the convenient data analysis and the visual presentation. If you're interested in our delivered content or you want to know how to use the system, I think you can visit this link about to register and to log in. Then you can use the demo data we provide to experience the convenient analysis of the Dr. Tom system. Okay. And if you visited the Dr. Tom system, I think you will find that besides the demo data for a single proteomics, for the single metabolomics, there also will have the data for the correlation analysis. So this is because the single omics can only provide the information from one part for our research subject. So while the multi-omics can help us understand the overall situation comprehensively. So to better help researchers to explore the sensitivity science questions in depth, we also design many multi-omics joint analysis services. Just showing here, we can provide the proteomics combined with different omics, such as the transcriptomics and the modified proteomics for the joint analysis. And earlier, we mentioned that in the single cell at a specific time, there is no resolution between mRNA and the protein levels. So therefore, to understand the phenotypes if you want to understand the phenotype more clearly, we can add the proteomics research and joint analysis for the proteomics and the transcriptomics are very important. And we will do some analyze the data from these several parts, the expression, the function, and the network and the interaction. And also we can do the transcription factor regulation analysis. So we can provide the various form of the analysis count and graduate, and we will deliver many rich image such as the network and the network interaction diagrams, and the pathway enrichment plots, and so on. So if using the joint analysis of the proteomics and the metabolomics can also help us explain the mechanism behind the phenotypes and better help us to discover the biomarkers. So besides the regular correlation, such as the network, the expression and function, the joint analysis of the proteomics and the metabolomics will also include the random forest analysis and the ROC curve evaluation. This will help us help the researchers find the biomarker more accurately. Okay, so that's all for today's webinar, and thank you very much for the watching, and I hope today's sharing will be helpful for your research.